This is lesson 9, chapter Principles of Inheritance and Variations. In today's video, you will learn about crossing over. The term crossing over that was introduced while discussing the types of linkage. That when crossing over takes place, we find incomplete linkage, and when there is no crossing over, then we find complete linkage. So this term crossing over, it have a relation with the inheritance of living organisms. As you know that the genes that are present in the chromosomes, they are arranged in linear fashion, and during the crosses, the combination of the parental genes, they appears to remain together generation after generation, and that we call it to be linkage. But this linkage, it is disturbed by a phenomenon which is called as crossing over. And this crossing over, this takes place during the meiosis cell division. So in this video, let's learn what is crossing over and how this crossing over takes place during the meiosis cell division. What is crossing over? It is the exchange of genes between non-sister chromatids of homologous chromosomes to produce recombinations or new combinations of genes during meiosis cell division. It takes place during Pachytene of Prophase I during meiosis cell division. The whole process of crossing over, it is a very simple concept that there the exchange of genes takes place, and this exchange of genes, it takes place between the homologous pair of chromosomes. Homologous pairs, that means the maternal and the paternal chromosome that have the characteristics to express a particular character, and that is present within a diploid cell. So this pair in a diploid cell, which is contributed by both the mother and the father, that is only called as homologous chromosome, and this homologous chromosome, they bears non-sister chromatid. And the crossing over, that is the phenomenon when the exchange of genes takes place between the non-sister chromatids of the homologous chromosomes, which as a result, it brings new combinations, or that in other words we call to be recombination, and this takes place during the meiosis cell division. As you know that in meiosis cell division there are many stages. So this crossing over, it takes place during the Prophase I of the cell division process, and in the Prophase I, it takes place in the Pachytene stage. Prophase I, that is again divided into 5 stages, and there the third stage, that is the Pachytene. And in that Pachytene stage, the crossing over between this non-sister chromatid takes place, where the exchange of genes takes place in between the maternal and the paternal chromosome to produce new combinations of genes before the formation of the gametes. So this is this very simple meaning of crossing over, that it is the exchange of genes between the non-sister chromatids of homologous chromosomes to produce recombinations. Now, what is the relationship between the linkage and recombination or crossing over? Linkage and recombination, they have a relationship, that is when the linkage is weak. As discussed earlier, that when the distance between two linked genes is more, there we find weak linkage. And when there is weak linkage, so there the chances of crossing over is more. So this is the relationship between the linkage and the recombination or crossing over. And in that case, always incomplete linkages takes place where new combinations or recombination of genes takes place in the progenies, and these recombinations, it brings about variation in the new offsprings. Now let's try to understand how this crossing over takes place during the meiosis cell division. The mechanism of crossing over, it completes with 4 steps. That is, first is the synapsis, then crossing over. After that comes the tetrad stage, and the 4th step, that is the terminalization of the chiasmata. As discussed earlier, that meiosis completes with 2 steps, that is meiosis I and meiosis II. And during the meiosis I, the first step, that is prophase I. This prophase I, this is also divided into 5 stages, and these are leptotin, zygotin, pachytene, diplotin, and dikinases. So this prophase I, this is a very significant step during the meiosis I of the cell division. And this crossing over process, it starts right from the zygotin stage. In the zygotin stage of prophase I, the single homologous chromosome, it comes to lie side by side with similar gene loci of the 2 chromosomes exactly opposite to each other, and this phenomenon is called as synapsis. In very simple words, when the homologous chromosomes, they pairs with each other and lie close to each other near the similar gene loci, as in the diagram you have seen that in the paternal chromosome gene A is there, and in the maternal chromosome gene capital A gene is there. And this capital A, they are the homozygous, they are in the homozygous condition, which is present in 2 different homologous chromosomes. Here they expresses the same character. So here during synapsis, these 2 similar genes which are present in the same locus of the homologous chromosome, they comes together, and this whole phenomenon, it is only called as synapsis, the coming together of the homologous chromosome, and it takes place during the zygotin stage of prophase I. Now next to the zygotin stage starts the Pachytene stage, and during this Pachytene stage of the prophase I of meiosis cell division, the crossing over, it starts. As you know, what is crossing over? That is the exchange of the genes in between the non-sister chromatid of the homologous chromosome. So here the single homologous chromosomes, they becomes 4-stranded, and there is the breakage of this non-sister chromatids where the exchange of the genes takes place, and later these non-sister chromatids, they again rejoin with their recombinant parts. So this is the second step of the mechanism of crossing over, where the exchange of genes, it gets complete. Now right after the Pachytene stage, the chromosomes, they do not separate completely. So there they have some points of attachments, and it takes place during the fourth step, that is diplotin stage of the prophase I, during the meiosis cell division. Here the synaptonymal complex that is formed during the zygotin stage, it begins to dissolve except in the region of the crossing over, and therefore the chromosomes, they separate, but the chromatids become distinct. And as we get the 4 chromatids now completely distinct with each other, but with the point of attachment, so this stage, it is called as the tetrad stage. Now during this tetrad stage, the attachment points between the non-sister chromatid of the homologous chromosomes, it forms the point of attachment, which is called as chiasmata. It is nothing, these are just the attachment points, the points of attachment after the phenomenon of crossing over that took place during the Pachytene stage. And this happens because right after crossing over, the homologous chromosomes, they do not separate directly. So here they uncoils with each other by opening the synaptonymal complex. Synaptonymal complex, that is a bridge that is formed in between the homologous chromosomes. And these homologous chromosomes, when the synaptonymal complex, it begins to dissolve after the phenomenon of crossing over, so there they forms this points of attachment, which is called as the chiasmata. And this opening of the chromosome after the crossing over, it gets completed during the dikinesis, which is the last step of prophase I. It gets completed during the dikinesis process, which is the last step where the non-sister chromatids, they start repelling with each other by the opening of the chiasmata, and this chiasmata, while opening up, it goes towards the terminal point of the non-sister chromatids. So after crossing over, these non-sister chromatids, they start to repel each other, and during dikinesis, these chromosomes, they detaches or they starts uncoiling from the nuclear envelope, and during that particular process, the chiasmata, they moves in a jeeper fashion. Jeeper fashion here means just like opening the jeep, as it goes from one end to the other end. So wherever these chiasma points are there, so they will start moving towards the terminal point, and this movement of the chiasmata is only called as terminalization of chiasmata. And as it reaches the terminal point, the two recombinant homologous chromosome, they becomes completely separate. So this mechanism of crossing over, it starts right from the zygotin of prophase I, and it gets completely it gets completed till the dikinesis of the prophase I stage. But the crossing over, it takes place right at the third step, that is the Pachytene stage. So this is how we get the crossing over process completed during the meiosis cell division, where we can say that shuffling of the genes takes place, or reshuffling of the genes takes place in between the maternal and the paternal chromosome to get different types of variations. And for this particular region, that we get so much variations in our nature. Now, why crossing over is important? Crossing over, it is important because it is a means of introducing new combination of genes and hence traits. As we know that in our offspring, the maternal and the paternal chromosome, they both contribute to express a particular character in the resultant offspring. Now, if there is no crossing over, then there won't be any new combination of genes, and if there is no combination of genes, then there will be no variation or variation of traits. So in order to get new combination of genes in each generation, this crossing over, it is important, and it takes place during the meiosis cell division process, during the time of formation of the gametes. Then the second point, that it increases variation, which is useful for natural selection. So you all have already studied about the evolutionary process, and during this evolutionary process, a very important concept, it was created, that variation is required, and for variation there is always recombination of genes. So this crossing over, it creates recombination of genes, and hence variation can be obtained. Then let's come to the third point, that it has proved that genes lie in a linear fashion in the chromosome. Yes, crossing over, it have proved that the genes, they always lie in a linear fashion in the chromosomes. And as discussed earlier, that the distance between these genes, that only determines the strength of the linkage. And if the linkage it is weak, then there is always chances of happening crossing over. And lastly, the useful recombinations produced by crossing over are picked up by breeders to develop useful new varieties of crop plants and animals. As this crossing over process, this is a natural process. Here, this is not an artificial process. It always takes place during the time of gamete formation in sexually reproducing organism. So these new combinations are only picked up by breeders to develop new varieties of either the crop plants or in case of animals. So these are the importance of crossing over. So there are also some factors that determines or that influence the crossing over process, and these are the distance. So distance between two genes, it determines both the strength of the linkage and the frequency of the crossing over between two genes. Then comes the temperature. So it also have a greater influence, because variation in temperature increases the frequency of crossing over. So temperature, it can play a very important role in the crossing over process. Then there are some other factors also, such as age, sex of an organism, as for example in case of the Drosophila. So there it was found that the phenomenon of crossing over in case of female Drosophila, it was quite common in them rather than the male. And lastly comes the heterochromatin fiber that is present inside the nucleus. So the presence of centromere and the heterochromatin areas decreases the rate of crossing over. So these are the few factors that influences the crossing over process. After discussing all these new phenomena about the inheritance pattern in case of living organisms, and especially those which reproduces through sexual means. So it is understood now that the genes, they are always present in linear order in a chromosome, and they are always linked, and they cannot be separated until when crossing over takes place. And it was found that crossing over, it is a very common phenomenon in case of the sexually reproducing organism during the time of formation of gamete. Now, under what condition does this law of independent assortment holds good, and why? Mendel, he was also not wrong during his experiment. He had also obtained the correct result from his experiment, that is the dihybrid cross ratio to be 9:3:3:1 and the monohybrid cross ratio to be 3:1. But according to the theory of linkage and the crossing over, so there the whole concept of the inheritance pattern, it got somewhat changed. Now let's see or let's find out the answer that how independent assortment it was correct, or how it holds good. Mendel, he was lucky enough to choose those contrasting characters which were present on different chromosomes. Therefore, the law of independent assortment it holds good to only those factors or genes which are located distantly on the same chromosome, or it occurs on completely different chromosomes. A chromosome bears hundreds of genes, and all these genes or factors present on a chromosome are inherited together, except the crossing over takes place in between them. So that is only concluded here that the linked genes, or the genes which are present in the same chromosome, they can also get assorted independently during the formation of gametes, as Mendel had said in his experiment. So he was lucky enough to choose those characters which were present in totally different chromosomes. Say for example, as you know that a pea plant, it bears 7 chromosomes, or the diploid set, that is 14 chromosomes. So out of that, chromosome 1 and chromosome 2, it bears the flower color and the seed shape. Similarly, chromosome 7, it bears the height of the plant. So in that way, those contrasting characters, that those were not present in the same chromosome, and therefore we can say that they are not linked gene. And here a condition is applied here, that independent assortment that holds good, that is when the linked genes, they are distantly arranged, that is when the linkage that is weak on the same chromosome, or it totally occurs on a separate chromosome. So in that case, Mendel, his law of independent assortment, it holds good. So this is all for the today's class. Thank you.